Welcome everyone. In this video, we will discuss the important questions from biotechnology principle and process. So let's begin. Question number 1. A researcher identifies a natural occurring variant possessing characteristics of interest. This plant is selectively bred. This is an example of a naturally occurring variant, which is a natural variation which plants naturally have, that will be selected by the researcher, and they will be do the selective breeding of that particular plant with that desirable traits. And this type of approach has been used for centuries to improve the crop yields, disease resistance, and other agronomically important traits. So it's a example of traditional plant breeding where a natural variation is selected and this selectively breeding will be taken place. So the correct answer is option A. Question number 2. Bacteria possessing restrictions endonucleases remain option A affected by bacteriophages, option B resistant to bacteriophages, option C resistant to drugs, option D resistant to heat. So the correct answer for this question is option B. Bacteria, those possess restriction endonucleases, they are resistant to bacteriophages because they have the ability to differentiate their own DNA from the foreign viral DNA. So they basically cut the viral DNA by methylation and protect their own DNA. So that's why those bacteria which have restriction endonucleases, they are resistant toward bacteriophages. Question number 3. Introduction of foreign genes for improving genotype is called option A biotechnology, option B tissue culture, option C genetic engineering, option D both A and B. So the study of foreign genes for improving genotype is called as genetic engineering, because in genetic engineering what we do is the alteration of DNA by cutting, inserting the foreign genes. So that's why the correct answer is option C genetic engineering. Question number 4. The liking of antibiotic resistant gene with plasmid vector became possible with option A DNA polymerase, option C DNA ligase, option B exonuclease, option D endonucleases. Let's look at every option. DNA polymerase, it is used for adding dNTPs for formation of DNA. Exonuclease, they used to cut the side fragments of DNA. Endonucleases, it will cut the specific site of DNA, while DNA ligase will insert that particular gene into the plasmid and it will behave like a glue. So the antibiotic resistance gene and plasmid vector will able to glued by this ligase enzyme. So the correct answer for this question is option C. Question number 5. Which of the following is the chemical knives of molecular biology are? So option A restriction endonucleases, option B exonuclease, option C reverse transcriptase, option D ligase. So the chemical life of a molecular biology is restriction endonuclease because it will only cut to the specific site of the DNA or the gene and insert the particular fragment there. So the restriction endonuclease is the chemical knife of a molecular biology. Question number 6. Plasmids are extra chromosomal genetic material found in option A algae, option B mammalian cell, option C bacteria, option D viruses. So, the correct answer for this question is option C bacteria. Plasmids are generally found in bacteria, but there are also some different organisms like yeast and fungi which have that plasmids in them, but the most common host for plasmids is bacteria. Question number 7. Which of the following bonds are formed by the action of DNA ligase? Option A sugar phosphate bond, option B phosphodiester, option C phosphate phosphate bond, option D both A and B. So the correct answer for this question is option D, both A and B, because the sugar phosphate bond is also called as the phosphodiester bond, as it is formed between the sugar and phosphate group in the nucleotide. So it's also called as the phosphodiester bond. And the DNA ligase is used to catalyze the formation of that phosphodiester bond between the 5' phosphate group and 3' hydroxyl group of another DNA strand. So DNA ligase form the phosphodiester bond, or we call it as a sugar phosphate bond. So the correct answer is option D. Question number 8. In recombinant DNA technology, a plasmid vector is cleaved by option A modified DNA ligase, option B a heated alkaline solution, option C the same enzyme that cleaves donor DNA, option D different enzyme that cleave the donor DNA. So in recombinant DNA technology, the plasmid vector is cleaved by the same enzyme that will cleave the donor DNA. So option C is the right answer for this question. The same enzyme that cleaves donor DNA will also cut the plasmid vector. Question number 9. Who is given the credit for constructing first artificial recombinant molecule? Option A. Hargobind Khurana. Option B. Stanley Cohen and Herbert Boyer. Option C. Linus Pauling. Option D. Arber and Nathans. So the correct answer for this question is option B. Stanley Cohen and Herbert Boyer were given the credit for constructing first artificial recombinant molecule. Question number 10. The specific DNA sequences where EcoR1 cuts is option A GA-TTCG, option B GAA-TTC, option C GTT-CAA, option D TTCCAA. So EcoR1 is a restriction enzyme that recognizes and cuts the specific DNA sequence, which is GAA-TTC, and it will form the sticky ends. So the correct answer for this question is option B. Question number 11. Identify the plasmid among the following. Option A HIND3, option B PBR322, option C lambda phage, option D both A and B. So let's look on each. HIN3 is a restriction enzyme, not a plasmid, so option A will be incorrect. Lambda phage is a bacteriophage, so option C is also incorrect. Only PBR322 is a plasmid vector, so option B is the correct answer for this question. Question number 12. Plasmids are important in biotechnology because they contain option A recognition sites on recombinant DNA strands, option B provirus incorporated into the host DNA, option C a vehicle for insertion of recombinant DNA into bacteria, option D surface for respiratory process in bacteria. So plasmids are generally used in biotechnology because they work as the vehicle for the insertion of recombinant DNA into bacteria. So the correct answer is option C. Question number 13. If gene of interest was inserted at SAL1 site in PBR322, the resulting plasmid will confer resistance to option A ampicillin, option B tetracycline, option C kanamycin, option D both A and C. So it's a bit confusing if you are not clear in this. So I'm explaining you for those who don't know what the question is asking. If we look in the plasmid vector PBR322, it have two sites, which is SAL1 and BAM-H1. This both are the restriction site present in PBR322. Now, when we will insert the gene of interest in SAL1 site, it will disrupt the function of gene that was originally present at that site. So, the SAL1 site is associated with the tetracycline resistance gene. If we insert the foreign DNA at this site, it will inactivate the tetracycline resistance gene. That means the bacteria will become sensitive toward tetracycline. And the ampicillin resistance gene is located on the opposite side of the plasmid and is not affected by the insertion of SAL1 site. So therefore, the bacteria will retain its ability to resist ampicillin. So the correct answer for this question will be option A ampicillin, because the insertion of gene of interest at the SAL1 site in PBR322 will confer the resistance to ampicillin, and they will be resistance toward ampicillin. Question number 14. Tumor inducing plasmid transforms option A nematodes, option B bacteria, option C fungi, option D several dicot plants. So the tumor inducing plasmids generally transform plant cells because the TI plasmids are found in soil bacterium known as Agrobacterium tumefaciens. When this bacteria infects a plant, it will transfer a piece of DNA from TI plasmid, which is called as tDNA, into the plant genome. And this transferred DNA will disrupt the plant's normal growth and development and leading to the formation of tumor, which is called as crown balls. So the correct answer for this question is option D. Question number 15. A bacterium commonly used in plant genetic engineering is option A. E. coli, option B. Agrobacterium, option C. Mycobacterium, option D. Rhizobium. So the bacteria which is commonly used in genetic engineering is Agrobacterium because they are found in soil and they have the unique ability to transfer a piece of their DNA, which is called as tDNA, into plant cells. So it's easy for the genetic engineering process to transfer the agrobacterium into plant for different changes in the plants. Question number 16. In RDT, the term vector refers to option A plasmids that can transfer foreign DNA into a living cell, option B plasmids that can cut DNA at specific bases, Option C, plasmids that can join DNA at specific bases. Option D, plasmids that can degrade harmful proteins. So, in recombinant DNA technology, vectors are referred as the plasmids that can transfer the foreign DNA into a living cell. So, the vectors are generally used as a vehicle to transfer the foreign DNA in the host cell. So, the correct option is option A. Question number 17. The tumor inducing capacity of dash is located in large extra chromosomal plasmid called TI plasmid. Choose the option which correctly fills up the blank dash. So option A is Thermus aquaticus, option B Salmonella typhimurium, option C E. coli, option D Agrobacterium tumefaciens. So the TI plasmid is called as tumor inducing plasmid, and it's a crucial piece of DNA which is found in bacterium Agrobacterium tumefaciens. So the correct answer for this question is option D. Question number 18. Genetic material of a retrovirus is option A DNA, option B RNA, option C protein, option D single stranded DNA. So the correct answer is option B RNA. Retrovirus have the RNA as its genetic material. So it's a basic question. You should know some basic questions also and learn more about the other type of genetic material present in which type of virus or bacteria or any microorganisms. Question number 19. Characteristics of vector include all except option A, presence of ori. Ori is origin of replication. So it's short called as ori. Option B, presence of antibiotic resistance gene as selectable marker. Option C, large size. Option D, MCS. So MCS is short form for multiple cloning site. If we look at the vector, it's have the ori region in it, means origin of replication is present in vector. They also have the antibiotic resistance genes and they use it as a selectable marker. They have the multiple cloning sites also, but they have the small size rather than large. So that's why the correct answer for this will be C, large size, because it's wrong. It should be small size here. It will be the correct characteristic feature for the vector. Why we don't see the vector in large size? Because generally they use as a vehicle to transport the gene. So if they have a large size, it's very impossible. But if they have the small size, they will be easily transferable into the different organisms. Question number 20. M13 can be classified as which kind of a vector? Option A, cosmid. Option B, YAC means yeast artificial chromosome, and option C, BAC, which means bacterial artificial chromosome. Option D, bacteriophage. So M13 is a cloning vector which is derived from the M13 bacteriophage. So the correct answer for this question is D. It's a bacteriophage type of vector. Now I have a question for you. Please tell me what is the genetic material of M13. Please answer it in the comment box.